Very good morning, dear children. Welcome back. This video is for class 10 science, chapter number 9, heredity and evolution, and this is video lecture number 2. In today's lecture, we are going to study the rules for the inheritance of traits, and we will see the Mendel's contribution in inheritance. So as we have to talk about the father of genetics today, so let us begin with the genetics first. What is genetics? Genetics is the branch of biology that deals with heredity and variation. It is also called the science of heredity and variation. Gregor Johann Mendel. He is known as the father of genetics because of the contributions he has given into the genetics. He was born in 1822 in Europe in a farmer's family, and he was educated. He studied science in mathematics in the University of Vienna. He discovered the principle of heredity by studying the inheritance of garden pea plant, which is called as Pisum sativum. He opted for this plant because it is having many contrasting characteristics, visible contrasting characteristics, and that is why he chose for the garden pea plant. Now, before going into the Mendel's experiment, let us first understand some of the basic important terms. And the important terms are chromosome. Chromosome is what? Chromosome is a thread-like structure inside the nucleus of a cell formed of DNA that carries the genes. Genes which are responsible for the characteristics. And the number of chromosomes, they vary in different organisms. Second is genes. A gene is a short length of DNA which codes for a protein. Genes are located on the chromosomes and they control various traits of an organism. Genes are the basic unit of inheritance and capable of determining the development of a specific characteristic of an organism. So, it is the gene basically which is responsible or which represents or gives the individual characteristics to the organism. The genes, they are represented by letters and they are always working in pairs. For example, if I talk of one characteristic, that is height. So, for example, we use a word T which says tallness. So, tallness is one characteristic and the genes, as we know, that they are in pairs. So one characteristic will be denoted by the letters, so capital T, and we will have small t in pair with it because capital T will represent the tallness of the plant and small t will represent the dwarfness of the plant. So both are representing the same characteristic, but they are working in pairs. These genes, they control the same characteristic. So they are controlling the height of the organism. So they are represented by the same letters. The genes, so here we have talked of the height, but genes that control the same characteristic, for example, here we have talked of the height. These can be classified into two different types: dominant and recessive. So about them we have studied in the previous lecture as well, that dominant gene is the one which decides the appearance of an organism irrespective of the presence of the alternate gene. That this dominant gene will show or will make the appearance, will decide the appearance of the organism, and it is not dependent on other gene. So it does not care for small t or recessive gene. So here smaller one will represent the recessive one and the capital one will represent the dominant gene. So the same genes, or you can say the genes that control the same characteristic, they into themselves can be classified as dominant and recessive one. And here the pair that we have, capital T and small t, they can also be called as allele. Allele means the factors that represent or that control the same characteristic. So capital T and small t are the alleles which represent or control the height of the plant. So here the recessive will decide the appearance of the organism only in the absence of the dominant one. When this will be absent, then only this recessive one will come into play. Then another term is genotype. Genotype refers to the genetic makeup, that how the basic design or the genes are working, or what genes are representing for an organism. So we can say genotype is the genetic makeup of an organism. It can also be explained as the combination of genes in organism. It is the genotype is also represented by the pair of letters. For example, here genes, a pair of genes was capital T and small t. So T, capital T represents the tallness of the plant. Now the genotype can be either capital T or T, capital T small t, or the for the dwarf plant it will be small t and small t only. Now the next term is phenotype. Phenotype refers to the physical or external appearance. So it says or it tells about external appearance of the organism. This is a visible trait of an organism. For example, we were talking of the height of the plant. So tallness. Tallness is one of the phenotype of the organism, as it is visible. So tall or short are the characteristics which are visible, so they will be called as phenotype. So those characteristics which are visible, they are called as phenotype, and which are making genetic relations, they are called as genotype. Phenotypes are always dependent on genotype. For example, if we talk of genotype capital T, T, one pair, or capital T, small t. Here, if we talk of the capital T and T, here both are capital, so both are dominant one and they represent the tallness of the plant. So in any case, this pair goes for the tallness of the plant. So the plant will be tall if genotype is capital T and T. Here also, if one capital T is present, which is dominant one, because capital T's are always dominant, so the plant will again be tall. If the genotype is small t and t, so this pair tells us that there is a pair of recessive recessive genes. So this will come into play as no dominant gene is there. So the plant will be short. So the short plant or the shortness of the plant, the dwarfness of the plant will be a phenotype, and this will be the genotype for the same. Now we have F1 and F2 generations. What is F1 and F2 generation? F1 and F2 both are filial generations. Here F1 filial generation, and so F1 will be the first filial generation and F2 will be the second filial generation. When two parents cross to produce offspring, let's say we have the male and the female, they cross together to form a new individual or the progeny, then this, because parent have crossed to make the new individual or the progeny, so this will be called as first filial generation. Now when the first generation progeny, this crosses over themselves, for example, if not just one, if we have sexual reproduction, then we can have one or two. So if we have two different organisms of the first generation, let's say we have another P. So when these two cross with each other, then the individual which is formed is called the F2 generation. So here the organism will be called as second filial generation or F2 generation. Now let us see what are the symbols which are used in the genetics. So generally letter symbols are used, or they were used by Mendel as well, to represent the different characteristics. So capital letters are used to represent genes for the dominant characteristics. For example, the capital T we used for tallness, we used it for showing the height, which was one of the characteristics of the organism. And small letter are used to represent the recessive characteristics or recessive genes. Now, any organism possesses two genes of each characteristic. For example, for the height only, every organism is having the dominant gene in it and the recessive gene in it. So, because it has got one from the male and one from the female, or you can say one from the father, one from the mother, no matter which one is from the father or mother. Now the next one is individuals having identical genes responsible for the specific characteristics is called homozygotes or homozygous. Homo means similar. So, if we say that from father also and from mother also, the dominant genes has come to the new individual or to the progeny, then this individual will be called as homozygote. This zygote which will be formed after getting these two genes of the same characteristic, this will be called as homozygote or homozygous. And the similar thing will happen if the recessive genes from the father and mother has come to the individual. So this will be called as homozygous only. But when from father or mother different genes of the same characteristic has come, for example the capital T and the smaller t, then this will be called as hetero heterozygote, and this will be homozygote. So now we have come to the main topic, that is Mendel's contribution in the genetics. So inheritance, that refers to the process by which the offspring acquire the traits from their parents. And each trait in the offspring can be influenced both by the mother and father, because we know that the cells, germ cells from mother and father combine to form a new individual in the case of sexual reproduction. So if we talk of Gregor Mendel's experiment, he made first discovery on the principles of heredity by studying the contrasting visible characteristics of garden pea plant. Garden pea plant was having large number of contrasting visible characteristics, so that is why he uh, chose for this plant. Now let us see what were the experiments done by the Gregor Mendel. So the first was monohybrid inheritance. First, Mendel investigated the inheritance of single contrasting trait. That means he chose only one characteristic to study. So mono means one, hybrid inheritance. So he studied the garden pea plant and its characteristics just by looking at one characteristic. So he considered only one characteristic and studied the plant, and that is why this experiment was called as the experiment of monohybrid inheritance. So Mendel, while starting with his experiment, he selected the plants with all the true breedings of the plants. That true breeding means either purely tall or purely dwarf. So for that, genotype will be TT and small tt. This will be the genotype for the true breeding of tall and dwarf plant. So this will be for tall plant and this will be for dwarf plant. So here, according to him, that tall plants progeny on self-fertilization produce the progeny of tall plants. That means when the tall plants they were self-crossed, or when they were crossed with the same plant as that of the same progeny, then they will always produce the plant of true breeding or the tall characteristic. And if the true dwarf plants are crossbred with the similar kind of the plant which is having the same genotype, then, or you can also call it as self pollination, then they will produce the plants which are having the true breed or true characteristic as dwarf only. So what did he do? That he crossed true breedings, tall plant. So here we have a true breeding tall plant and true breeding dwarf plant. And when they crossed, or the pollination between a tall and a dwarf, true tall and true dwarf plant happened, then what are the possibilities? Actually, Mendel was the one who brought the mathematics, probability mainly, into the biology. So what happened? If we see here, then we can check what are the various probabilities. Suppose these two are the same factors of the gene, or the genes of, or a pair of gene which represent the same characteristic. So if these two cross with each other, then there are possibilities that from here the T can come, from here also the first pair can come and form TT. From here also the possibilities are capital T is there, and let's say from here also second comes and we get second small t. Now there is a chance that first T can also meet with the second T of this pair, and here also the second can meet with the first one, but always you have to write in the same manner. First the capital one will be written and then the smaller one will be written. So here again we will have the capital T and T. So here if we see, then we know capital T is the dominant gene and the smaller t is the recessive gene. So finally what we are going to get, a tall plant. In all cases, in all probabilities, tall plants will be produced. Why so? Because till the time when dominant gene is here, then only that will make the physical appearance of the plant. So all tall plants will be produced. So when these two true breedings crossed, that was parental generation, and this, when we have got all the tall plants, this is called as first filial generation or F1 generation. Now further there are chances that many things can happen in further crossings, but here we can see, if we talk about the genotype here, so we know what were the various genotypes. The main genotypes are the capital TT, small tt, and capital T small t. The genotypic ratio was, we were having one pair of both dominant, one pair of both recessive, and we have in total two more pairs from each of the uh, pair which is having one true or the dominant one, and one is having the, here in total four cases what we have, two main points have been concluded, that here the genotypic ratio will be that we have one true breeding of tall, then two more breedings of the tall one, and one for the dwarf one. What is the basic phenotype here? Either tall or dwarf. Now, what can be concluded on the basis of this experiment? So here you can see that the same thing Mendel observed, that the dwarf trait of the parent pea plant which was absent in F1 generation here, because here no dwarfness has come. This will reappear into the second generation. How? Because there are chances when the two, let's say we have capital T and the small t, and we have the capital T and the small t. They cross with each other, any of the two, then we will have chances, if the first generation cross with each other, then second generation will come. So here we are having chances that first can meet with the tall and with the smaller one. So we will have capital T T, capital T small t. Second can go also go for the same, then we will write capital T first and then this second one, and then we can also have one pair which will have small t and t. So this is the second filial generation. Now this shows that the dwarfness, which was one of the characteristics of the organism, that has not been lost, but it reappeared in the second generation. So Mendel called the suppressed feature, because this was the suppressed feature in generation first, as the recessive trait, and expressed the feature of tallness as the dominant one. So according to him, each of the pair which we were having, the tallness, the true tall feature and the true dwarf feature, they remain segregated. They are not lost while crossing with either self pollination or cross pollination. So they remain segregated from each other. They do not blend with each other. So we can conclude that according to Mandel's, genes controls the trait. So it is actually the gene which controls the characteristics, whether as for example tallness or shortness. And the second conclusion is that internal factors means the same or the type of genes which represent same kind of characteristic. Occurring in pair, they determine the traits of the organism. So, for example, we had capital T and T. So, they determine the traits of the organism. Whether the new progeny will be tall or dwarf, it is decided by this pair of internal factors. Factors are basically the genes, which were later known as genes. Earlier, they were called as factors. And a single gamete consists of only one of a pair. So, single gamete, the cell from father, will carry either one of them, and from the mother, the sex cell which combine together to form a zygote. Each of the cell has either of them, and hence in final zygote we will get the two, each from the one from the each. So this is called as law of segregation. The second experiment was dihybrid inheritance, the study of two characteristics at one time. So here, this time, Mandel took shape and color of the garden pea plant at a time and studied it. So, shape and color were two pairs of contrasting traits because these are the visible ones, which are visible ones, and Mendel chose them because they are contrasting. And the garden pea plant is having the variety in that. So, what did he do? He crossed pure breedings of pea plants which are having, because shape can be round, or let's say capital R is the round, and another type can be wrinkled. Color can be yellow or green. So the garden pea plant was available in the two, so that is why he chose for the garden pea plant. And what did he do? That he crossed pure breeding pea plants having round seeds with the yellow breeding, and the wrinkled one with the green seeds. So, all the pea plants produced in first generation were round yellow seeds, round yellow seeds, and so let us see how does it happen. So, here in the experiment, what do we have? We have one round yellow seed, another is wrinkled green seed. Remember, round yellow, wrinkled green. Now, when these two cross with each other, what has formed? A round yellow seed has formed. Round yellow seed. So, here this was our first filial generation. When round yellow seeds were selfed or they were crossed with the same type, then what happened? Then there were possibilities that round yellow seed will form, round green seed will form, wrinkled small r, wrinkled yellow seeds, or wrinkled green seeds. So here we obtained when first generation crossed with itself, then there are possibilities that four different kinds of uh, organisms can form in the second generation. So here, what do we obtain? That the characters while crossing with each other are not getting lost here, but they. So here we can clearly see that when we were having the round green seed and the wrinkled yellow, when these two crossed, what we had, we were having round yellow and wrinkled yellow. Then further they formed a round yellow seed. So there are possibilities to form the capital R and small r, capital Y and small y. Now we have to check the variance or the possibilities that can happen. There are things that if this is the first generation, now if it crosses or it is crossed pollinated with itself or self, then what are the chances? So here we can have that, what do we have? The possibilities are round yellow, wrinkled yellow or wrinkled green. So, and here also the same chances are there. So if we see, we write them here and we write the same thing here. Now what are the possibilities that we can have? The progenies can be, that can be either round yellow, both from both, round can come from the uh, shape, and from the color yellow can come. What else? Here round yellow, and the second yellow can be the recessive one. So here are the various possibilities that we can have in the form of the genes which represent the same characteristic. Here you can see capital R and capital R are representing the dominant gene for the shape round. Capital Y, dominant gene for the color yellow. So here one pair is of the color, one pair is of the one pair is of the shape and one for the color. So here the various possibilities that we can see are these. So in conclusions we can say that although the combination of two pairs of original traits, we were having the shape and the color. So for the color we can have either, for color itself we have either yellow or green. So for yellow we will have the capital Y and the small y. For green we will have the capital G and small g. Similarly for shape we have round and wrinkled. So for round we will have capital R and R. For wrinkled we will have the, if we write it as W, then capital W and small w. So these can be the genes with the which represent the same characteristic. Here this is representing the red color, round shape. This is representing the wrinkled shape. Capital Y is representing the yellow color. Sorry, capital Y and small y. Then green, capital G and small g is representing the green colored characters or the genes. So here he concluded that the combination of two pairs of original traits, pairs of. So here two pairs of original traits combine or combine together, but these pairs they separate in the successive generations and they behave independently, that we have seen in the flow chart of it. So here we can say that although they combine together but they separate further in the next generation. So that is why this law was called as law of independent assortment. That means pair or pair of genes which represent a particular characteristic do not blend with each other, even though if they combine with other pairs or the different characteristic pairs, then also they remain independent and they keep on separating or behave independently in the next generations. So, Mendel's work was concluded in the form of three laws, and they're called as Mendel's law of genetics. First law is called as law of dominance. This law says that when the crossing between the organism occur for contrasting traits, only one character of the pair expresses itself in the first generation, and that the character that expresses itself is called the dominant one. For example, we talked of the capital T and the T. So they represent height both, but the capital T will be the one which will represent it whenever the smaller t is present in with them. And the smaller t does not represent it until the capital T is remaining with it. So that is why this is called as recessive gene, or uh, the other one will be called as dominant one. Then we have the law of segregation. So this law says that genes stay together, segregate at time of gamete formation. That means the genes, they remain together, but when the zygotes are formed, that time they separate or segregate from each other. This means the mixing of genes in the form of hybrids does not occur. That means the blending of different characteristics does not happen. They remain individual and independent. This is also called as law of purity of gametes. Now, the another one is called as law of independent assortment. We have just seen that when crossing occurs between the individual differing in two or more than two pairs of contrasting characters, the inheritance of any one pair is not affected by the presence of the other. For example, the inheritance of tall characters of the plant is not related to the smooth characteristics of the seed. So, the shape and the size both are different characteristics and they do not mix with each other. They remain independent in the generations. So, these two characters are inherited independently. So, this was all about today's lecture. Now, let us see today's homework. So, children, in today's lecture, we have studied about Mendel's experiment. So, we will be doing the in-text questions at page number 147. Here, question 1 and 2 of this page number 147, are to be done as the first two questions of the homework of lecture number 2. Question 1 is, how do Mendel's experiments show that traits may be dominant or recessive? Question number 2 is, how do Mendel's experiments show that traits are inherited independently? So here you have to explain the law of independence, or law of independent assortment, and here you have to explain the law of the dominance. Question number 3 of the homework is, what is the difference between homozygote and heterozygote? Question number 4 is, what does the law of independent assortment state? So these are the 4 questions which you are supposed to do in your fair notebooks. And children, the homework solutions of lecture number 1 will be discussed in the online class. So we will see you in the next lecture, in which we will learn the sex determination of an organism. So till then, take care and goodbye.